let me go to slideshow. Okay, so any questions on this so far? I haven't. If you have any questions, you can put in the chat box. Okay, otherwise I'll continue with the class. So what we have done, in the last class, and even just now, is study the action potential generation as shown in the Hodgkin-Huxley model. So Hodgkin-Huxley model represents a small patch of the neural membrane, right? And so in that patch, if you just inject some current, how does the voltage change? That's what we have discussed using an electrical equivalent circuit. So now what we'll do is we'll go deeper. We will proceed in two different directions over the next few weeks. Right. One is we look at more detailed models than even Hodgkin-Huxley model. Because Hodgkin-Huxley model basically talks about how the action potential is generated near the axon hillock or near the cell body. So if you can think of cell body as a sphere, right? If you inject some current into that sphere, how does the voltage of the membrane change? That's what it looks at. So it is basically looking at a neuron as a ball, as a kind of a point unit. In fact, such neuron models are called point neuron models. But neurons are extended structures. They have lots of wires coming out, and they have lengths and thicknesses and so on and so forth. So if you want to model a neuron in greater detail, then you have to explain and model exactly what happens in the various cables of the neuron, what happens in the dendrite, what happens in the axons, and what happens at the synapse, right? That's a model of an extended neuron, which is extended in space. So we'll from the current position where we have modeled as the neuron as a point in space and voltage change at this point, we'll now look at extended models of neurons, where we look at the signaling components in the extended body of the neuron. So there you have several signaling components: dendritic processing, axonal processing, and action potential generation is something that we've already looked at. That's the Hodgkin-Huxley model, and then synaptic transmission. Okay, so that is, we'll discuss that, and you can see that this kind of modeling can get very intricate, very detailed, quite involved. So we'll see that for some time and understand some basics. We'll also look at a software called Neuron. It's a simulator which will help you simulate neuron at this level of detail. So then we'll kind of start climbing down. All right, then we'll look at models which are much simpler than Hodgkin-Huxley model. So the upper box represents models which are more complex than Hodgkin-Huxley, and the lower box represents which are much simpler than Hodgkin-Huxley. Hodgkin-Huxley has 4 variables. We'll look at models of 2 variables and 1 variable and no variable at all. That is constant neuron models, right? And then using such simple neuron models, we'll start building circuit models or network models. Okay, so this is an example of biophysical modeling. So basically, you take the arbor of the neuron, right, with its the cables, with its branches and everything, and express them as electrical equations and electrical circuits, and describe the signal propagation in such circuits. Okay, this is what is called biophysical modeling. Then for the simplified neuron models, we look at 2 variable neuron models. There are many of them, like for example, Fujinakumo model, Morris-Leclerc model, and so on and so forth. So in this case, you can use your excellent phase plane techniques. You can draw null planes, you can find out fixed points, and so on and so forth. All those methods we will use there. Okay, so this is a kind of a overview, right, of things to come, right, over the next few weeks. Now let us look at this biophysical models of a neuron. We look at all the 4 signaling components. So as you remember, there are 4 signaling components in neuron: the dendritic processing, and then the summation in the neuron, right, and the action potential generation at the axon hillock. Then the action potential propagation along the axon, which is axonal propagation. Then once signal reaches the end, then you have synaptic transmission. So you have dendritic processing, action potential generation at the axon hillock, axonal processing of the action potentials, right, where it propagates without loss of amplitude, and then neurotransmission at the synapse, right, where a chemical released by the presynaptic side is converted into a voltage on the postsynaptic side. Okay, so you see that this whole sequence occurs in a unidirectional fashion from the dendrite to the axon terminals. So let us talk about dendritic processing, the first step. So we have just verbally mentioned before that in the dendrite, the propagation is passive. So therefore, if you inject some current somewhere in the dendrite and produce a voltage wave, that voltage wave as it propagates, it loses amplitude and expands in time, right? Spreads in time because there's no energy, because it just keeps losing energy and it doesn't get recharged as it propagates, like unlike what happens in the axon. So if you look at the circuit model of what happens at a single point in the membrane, right, or a small patch of the membrane, this is what we have derived in the last class. This is Hodgson-Sley model. Only thing is here you have these active elements, these voltage sensitive channels, which make the whole circuit nonlinear, and it is through these voltage sensitive channels or through the form of these nonlinear elements that the circuit becomes an active circuit. That is, these are able to source energy. So there's a lot of biochemistry behind it, why it happens and all that. I won't talk about it now. But so that's how they're able to source energy, and therefore the circuit is able to produce this kind of amplify the brief input, right? A small current input and produce a large voltage response. So energy is getting injected in the form of these nonlinear conductances. Okay, but on the dendritic side, it is passive, right? And there is no voltage dependent channels. So therefore, the circuit can be simplified further if you are trying to develop a similar circuit for the dendritic side. This circuit, which was meant for the more like the axon hillock or axon side, right, can be simplified further because on the dendritic side, typically there are no voltage sensitive channels. But again, in biology, there are no absolute rules. For every rule, there's an exception. But I don't want to confuse the picture right now. I'll give a more mixed picture later on, but let me give a clean, simplistic picture right now. Otherwise, it's too confusing, right? So on the dendritic side, there are only passive channels, and let me say that right now. And therefore, this picture can be simplified further because if these are not voltage sensitive channels, then I can remove the arrows. Then you will just have three branches consisting of conductances and voltages. And using Thevenin's theorem, you can combine all three branches and come up with only one branch of conductance and voltage. So then it looks something like this, right? So you have capacitor, and then you have the conductance and battery combination, series combination. And this will be the circuit model at a given point or a small patch of the membrane on the dendritic side because it's all passive. But this is still a small point at a given point or a small patch. But if you want to model the entire length of the dendrite, obviously this is not enough. What you need is you will have to take many such units, each representing one small patch or one small segment of the dendrite, and then connect them using resistors because when current flows through the dendrite, it is resisted. There's some resistance to the current there because it's a real world. It's not a superconductor or anything. So there is some resistance there. So this is basically a good model of good electrical model of signaling inside a dendrite. So let us develop this idea further and formulate what is called a cable equation. So this part of the course, this part of discussion, the reference for this is Christoph Koch's book called Biophysical Computation. I think it's there in the library, but anyway, my notes, which is a web notes available, and it covers all these things. You can look at that. So now you see that in this equivalent circuit, you have the capacitance, then you have the conductance here, and then the resistance, which is along the length of the length of the dendrite. So the first element that we have to define is this resistance, which is located along the length of the dendrite, and that's called the axial resistance. So this is a resistance offered by the intercellular compartment, and it's defined as per unit length. So because when you write down the equation, you will write down the equation at a given point in the cable. It will be a partial differential equation where the independent variables are both time and space. Space means length of the cable. So we'll define this resistance in terms of per unit length of the cable. And we assume that the cable is of diameter d, and the resistivity of the intercellular medium, which is the basis of this resistance, is denoted by R sub I. Now this R sub I is the notation used in this neural cable equation context. But if you look at your high school electric circuit theory, resistivity is denoted by the symbol rho, right? It's exactly the same quantity. Only thing is the notation here is Ri. So this resistivity Ri, which is an intrinsic property of the material of the internal material, the intracellular medium of the cable, we relate this to the axial resistance R sub A, which is a resistance per unit length of the cable. So how do we do that? So we know that from your high school, electric circuit theory, that resistance is equal to rho L by A. Rho is the resistivity, R is the resistance, L is the length, and A is the cross-section area. So if you take a cable of length L equal to 1 unit length, then it will simply be rho by A. Okay, only thing is here we'll use the notation Ri instead of rho. So therefore you have so Ra, right, this axial resistance, which is defined as resistance per unit length, is given as Ri by A, just like rho by A. Here you have Ri by A, and so that's Ri by pi d square by 4 because diameter of the cable is given. So you have this quantity, and this is resistance per unit length. Therefore, we denote it by the units ohms per centimeter. So schematically, you can denote this resistance like this, right? It is along the length of the cable. Now let us define another quantity called membrane resistance. So membrane offers resistance for the flow of current between inside of the membrane or inside of the cable and outside extracellular space, right? From between the intercellular compartment and extracellular space. So this resistance is inversely proportional to the surface area of the membrane. Because here ideally you should, it's more convenient to talk in terms of conductance because the resistance is offered by the membrane, and then that resistance will be lower if the area is higher. So therefore, if you talk in terms of conductance, the conductance will be higher with area is higher, so because proportional to area. So, but the notation in the textbook uses resistance. We'll talk about resistance, but we'll relate to the conductance. So now capital Rm is the resistance of the membrane patch of unit area, all right, and the quantity is referred to as a specific resistance of the membrane, and the total resistance offered by a cylinder of diameter, right, d and of unit length L is given by. So if now small gm, let us say, is the conductance of this of a membrane of unit length or of length L, then gm is the conductance of membrane of unit area, right? So then obviously the conductance of that segment, that cylinder, will be equal to gm times area of the cylinder, which is equal to gm times pi d, which is circumference, times length of the cylinder. Now the small gm is given by 1 by Rm because by definition, and capital Gm is also given by 1 by capital Rm by definition. So therefore, you can just rewrite these equations as small Rm is equal to capital Rm by A, which is equal to capital Rm by pi d into 1. The length is 1, right? So therefore, it is equal to Rm by pi d. And so what we are saying is the resistance of a membrane of unit length, right, is given by capital Rm by pi d, where capital Rm is the resistance of the membrane of a unit patch of the membrane. So units of small Rm is ohm centimeters. Okay, so then the third quantity we have to define is membrane capacitance, because membrane also has a capacitance, right? And the specific capacitance, capital Cm of membrane, of a neural membrane, is typically about, because this is a property of the material of the membrane. It doesn't have anything to do with its lengths and all that. So it's about 10 power minus 6 farads, that is my 1 microfarad per centimeter square. Therefore, the capacitance of a cable of unit length Cm, just because the arguments are very similar to the previous case. In the previous case, also conductance is proportional to the area. Here also capacitance is proportional to the area. So you have very similar arguments. So therefore, here you have small Cm is equal to uppercase Cm times pi d, which is given as a farad per centimeter. So now with all these basics, you can represent a cable which is a passive cable, which is typically like a dendrite, as an electrical circuit. Right, we can put all these previous elements, that is a small RA, small RM, right, and small CM. We can put them in a circuit model which looks like this. This is similar to circuit we just defined a few slides ago. So the at a single point, I just have RM and CM, right? But so I have now consider 2 points which are apart by a distance of about delta x. Then you have to take two such circuits and connect the two circuits using a resistor, which is the axial resistance. Now you would be wondering what happened to the battery. So what we'll do is since both the circuits will have a battery of the same value of what is called the V rest, is the resting potential, right, of the membrane. And what we'll do later on, we'll keep it, but we'll redefine the membrane voltage so that we include this resting potential. So we do like a coordinate transformation, okay, and get rid of the battery by redefining the voltage, which will take care of that baseline voltage. Therefore, you just kind of understand that this is included. It will be taken care of later, so we'll remove it right now. So this is the circuit equivalent of the passive cable, which is dendrite. So input current here is axial current, which is II. Current here is at x minus delta x. Current here is II at x. This is here it is II at x plus delta x. Now the membrane currents is denoted by Im. So here it is Im of x, and here it is Im of x plus delta x. The axial resistance of this segment is Ra times delta x. So Ra here. And then the membrane resistance is Rm and capacitance is CL. So let us look at how these quantities are related or relatable. So if you take this stretch of the cable, length of the cable, and just apply Ohm's law, what do you get? The current going, so if you look at this segment, current going in to this cable is II at x minus delta x. Current here is II at x, right? And and the voltage here is V of x, and voltage here is V of x plus delta x. Right, and current here is II of x. And if you apply Ohm's law here, Vm at x minus Vm at x plus delta x, okay, is equal to basically IR. So I is II, and R, this Ra is only the resistance per unit length. So to get the actual resistance of a cable whose length is delta x, you have to multiply it with delta x. So this is the resistance, and that times II. So this is IR term. So if you take delta x to the left side and take the limit to 0, you get dou Vm by dou x is equal to Ra into II at x comma t. This is the first equation. Now apply the Kirchhoff's current law, like current conservation law at the nodes. You have two nodes there. Take any node and apply it. So what you get here is the input current here is II of x, okay, and so the II of x is split into Im of x and II of. So II of x minus delta x is split into Im and Im at x and II at x. So let us look at that. So II at xt is minus II at x minus delta t, delta x comma t is equal to minus delta x Im xt. So Im also is a current density, right? If you want to know the current going through a cable of length delta x, you need to multiply it with the actual length of the cable, which is delta x. So now if you take delta x to the other side, you get dou II minus dou II by dou x is equal to Im of x. So basically derivative, the spatial derivative of the axial current gives you the membrane current. That's what it says.
Yes, sir. One question
Yeah.
here.
Yeah.
So this Im is the current density, and II x minus delta x is not a current density, right? That is why we are not multiplying by the delta x factor there.
Yeah, II is the current. II is not. II is the total current going through the cable.
Correct. Only the Im is the density.
Im is the current density.
Okay.
Yeah. So was there a question on that?
No, because we are multiplying the delta x factor only to the Im.
Yes. And similarly for the axial resistance.
Yeah.
this we have to calculate, multiply RA, which is the resistance of a unit length of the cable,
Correct.
right? Because its length, this units is ohms per centimeter.
Yes.
Okay, and that is multiplied by delta X to get the actual resistance. So if we
Okay.
apply Ohm's law, the voltage difference between here and here times RA times delta X. I mean, so which is equal to RA times delta X times current, which is IA. So IA is amperes.
Okay.
IM is is per unit length.
Yeah, amperes per centimeter square, probably.
Per centimeter.
Yeah. Okay.
No, because the thing is, earlier we used to define conductances per unit area, because for Austin-Huxley model we were discussing what happens in a patch of the membrane.
Okay.
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 And the basic quantities are also defined in terms of the per unit area. But when you are describing the variation for a cable, right? In a cable, we are only interested in what happens along the length of the cable. We are
That's
not
right.
interested in what happens in the cross section of the cable. So we assume that within the cross section, everything is uniform.
Okay. Okay.
So we don't want to look at the cross sectional details and the diameter and all that. Right? So basically, what we have done in the last several slides, actually good that you asked this question. In the last several slides, we defined axial resistance, defined the membrane resistance, all that. Basically, what we were doing here is define quantities. So from quantities which are defined in terms of per unit area, we define quantities which are defined in terms of per unit length, right? Because they are more convenient to describe dynamics on a cable. Because in a cable, we assume that only the linear dimension, the length of the cable is important. Others are all assumed to be zero or negligible.
Okay. Okay.
Okay, so all this redefinition of quantities is required just so that you define quantities in terms of per unit length.
Okay.
All this technical subtleties arise only because we are defining everything in per unit length.
Right. Yeah.
Okay.
Yes.
So yeah, good that you asked that question. So now you have the second equation is now dou I by dou X, right? So IM is amperes per or microamps or whatever, amperes per unit length per centimeter. So therefore, dou II by dou X is equal to IM. With the second equation. So if you combine the first equation, so II is given here. So if you combine these two, you get 1 by RA dou square Vm by dou X square is equal to IM of X. So just plug this into this. Okay, you get this. That is first thing. So then what is IM? So because IM we just said we just said IM, but we haven't expanded it. The IM has several components. There's some current going through the resistance RM, some current going through the capacitance CM. So we have to split those components. So IM can be resolved into three components actually. There's the current. So basically the currents that are kind of vertical, okay, going across the cable, crossing the cable membrane. Right, there are actually three such currents. One is the capacitance current. Then they have the membrane resistance current, the RM current going through the RM. The third is if there is any external current, right, which is injected from outside. Suppose in experiment, you're doing an experiment like Hodgkin and Huxley, right? They poke the axon with an electrode and inject current. Okay, that is IX. Or when there is a synapse, right, of another axon, another axon synapsing at a given point in dendrite, when it releases neurotransmitter, that release of neurotransmitter effectively injects some current into the dendrite. That's why there is a post-synaptic potential. Right. So this is another example of a current which crosses the membrane from outside, not because of internal factors, but external agency injecting current. So these are the three different kinds of current which cross the membrane. So we need to accommodate all this. Okay, so all this together we'll call it the IM. So the resistive current, right, current through the RM is simply given as Vm minus Vrest by RM. Okay, and the capacitive current is given as Cm dou Vm by dou T, and external current is given as minus Ix, because these two are outgoing currents, right? Vm minus Vrest by. So here we are note that we are using that Vrest, that is the Nernst potential that we had in series with the conductance. That we didn't drop it, we didn't just ignore it or anything. It is shown here. Vm is a membrane current, membrane voltage, and Vs is the resonance potential of the membrane. So we have taken the difference by Rm. There's a right. Yeah. So note that this is all per unit length quantities, right? So because Im itself is per unit length quantities, all these are per unit length quantities. So that the units is amperes per centimeter. Now combining, so this is how we define the Im. Im is all currents that cross the membrane, right? II is currents that go through the membrane. Im is currents that cross the membrane. So whenever you have something that crosses the membrane, you have to talk in terms of per unit length. Okay, so plug in that value here in equation 3. So you get a more comprehensive equation, complete equation. So this quantity 1 by Ra times dou square Vm by dou x square is equal to this whole thing. Okay, and here what we are doing is multiply both sides by Rm. Okay, so you get and redefine and introduce two new terms. One is tau m, which is defined as Rm times Cm. Another is lambda square, which is defined as Rm by Ra. Okay, so if you do that, the same equation, and if you do that and combine equations 3 and 4, you get this equation, right? Which is lambda square times rho square and dou square Vm by dou t square is equal to Vm minus V rest plus tau m times dou Vm by dou t minus Rm Ix. So now we'll redefine the membrane voltage as the difference between Vm minus V rest. We'll say that is the membrane voltage. Then in the derivatives, it doesn't show up. It doesn't show up in the Ix current. So we'll simply have the same equation without that V rest term. Only thing is we understand implicitly that the Vm here means the actual voltage of the membrane minus V rest, whatever be the resonance potential. So this equation is called the cable equation. And here we introduce two new terms, two new parameters. One is called lambda. Lambda is defined as square root of Rm by Ra. And note that Rm by Ra, the ratio, the square root of that unit, the units of that quantity is length. So lambda is called the space constant, right? And tau m is Rm times Cm. The units of this quantity is time. Therefore, this is called time constant of the cable. So basically, to give an intuitive idea of what these quantities means, right? If I inject some current, we said that the wave as it propagates, it loses amplitude, right? So how, if you ask like, how much distance does it have to travel before it loses amplitude by a certain factor? Suppose you ask that question, right? Then you are invoking the notion of some kind of a space constant. So similarly, if you inject current at some point, it will produce a huge voltage wave at that point immediately, right? If we inject a pulse current, we will produce a huge voltage wave immediately at that point in the membrane. But after some time, since that current gets dissipated, it flows in both directions, that voltage wave reduces in amplitude. So now if you ask the question, how long do you have to wait before my initial voltage wave has to drop by 50% or something like that? Right, to answer that question, you are invoking some kind of a time constant, some notion of a time constant of the membrane. Right, so both tau m and lambda exactly satisfy that need, because you can see that it is the membrane has certain temporal characteristics, right, because of the capacitance, resistance, and all that, and spatial characteristics because it's extended in space, right. These two are captured by these two quantities, tau m and lambda. So this is called the cable equation. Okay, and so we will discuss how to analyze this, how to solve this. It's pretty hard equation to solve, but we'll look at initially steady state conditions, which is much easier to analyze. And then slowly we'll look at full temporal solutions of the equation and see how we can apply it to dendritic propagation. So thank you. Let us do that in the next class. That is